Liver cancer, known medically as hepatocellular carcinoma, is one of the leading causes of cancer-related death worldwide. It often develops in people with chronic liver disease or fatty liver. But beyond inflammation and genetics, cancer cells also depend heavily on metabolism. Tumors must rewire how they process nutrients in order to survive and grow. Researchers focused on a particular class of fats called ether-lipids. Lipids are essential components of cell membranes, but when certain lipids accumulate abnormally, they may influence tumor behavior. The team asked, could ether-lipid buildup actively promote liver cancer progression? The study identified a key factor, PPAR-alpha. PPAR-alpha is a protein that helps regulate lipid metabolism in the liver. Under normal conditions, it maintains balance in how fats are processed. But when PPAR-alpha activity is reduced, lipid metabolism shifts. In liver cancer models, researchers found that reduced PPAR-alpha activity was linked to increased accumulation of ether-lipids. This buildup altered cell membranes and signaling pathways. The result was increased tumor growth. In simple terms, a disrupted metabolic regulator allowed certain fats to accumulate, and those fats supported cancer development. Further analysis showed that ether-lipid buildup influenced oxidative stress and survival signaling in tumor cells. These changes created a microenvironment that supports cancer cell survival. Importantly, restoring or activating PPAR-alpha reduced ether-lipid levels and slowed tumor progression in experimental models. Cancer metabolism is an expanding field of research. Beyond genes that drive cell division, scientists are examining how altered nutrient processing supports tumors. This study highlights ether-lipids as potential contributors to liver cancer progression. It also positions PPAR-alpha as a possible therapeutic target. This research does not suggest that dietary fat alone causes liver cancer, but it demonstrates that how cells manage lipids internally can influence tumor behavior. As liver cancer incidence rises globally, especially with metabolic disease, understanding these lipid-driven pathways becomes increasingly important. Future clinical research will determine whether targeting ether-lipid metabolism can complement existing therapies. Cancer is not driven only by genetic mutations. It is also shaped by metabolic shifts that may open new treatment strategies.